Hi everyone, my name is Derek, and today I'm very excited to be presenting our work on computational modeling of the germinal center response using Computational 3D. So today I'll start with some biological ideas behind the germinal center reaction. Then I'll review our Computational 3D implementation and do a quick walkthrough of the code. Next, I will talk about our Python code for exporting and visualizing data that was generated from the simulation. And finally, there will be some exercises for everyone to do in terms of plotting the simulation outputs. And this will be outside of Computational 3D, but still in Python. So to start, I'll be going over the biological processes of germinal center responses and why they are so important to our immune systems. So there are two main aspects of the human immune system, with the first being innate immunity. And this includes external defenses like the skin, and they form the first line of defense. The second line of defense is adaptive immunity, and this involves immune cells targeting specific antigens using antibodies, so humoral. And the majority of defenses against pathogens come from adaptive immunity. And so in adaptive immunity, there may be T-cell independent or T-cell dependent humoral immune responses. T-cell dependent responses are the ones that produce memory B cells and a long-lived response. So that's what we're going to be focusing on. So T-cell dependent humoral immune responses take place in locations called germinal centers, and they're basically where B cells undergo maturation. B cells produced in germinal center reactions are affinity matured, which means high affinity antibodies that can quickly bind to antigens. And upon exiting the germinal center, they become long-lived memory B cells or high affinity antibody secreting plasma cells. And during secondary exposure to the same antigen, these memory B cells can allow for fast proliferation of these plasma cells, which is very important. So going over the structure of germinal centers, which is where GCRs occur, the germinal centers are essentially specialized microstructures that form in B cell follicles of secondary lymphoid tissues. And this is in response to infection or immunization. And for a typical secondary immune organ like the lymph node seen here on the left, GCR occurs in secondary follicles in the outer cortex. So that would be right here. And then zooming in on the germinal center here, you can see that it's distinctly round-shaped, and there are two zones. The dark zone, which is shaped like a waxing moon, is more populated, which is why it's darker, and it's packed with more proliferating B cells. And the light zone here is shaped like a waning moon, and it's less populated and filled with non-proliferating B cells. And in this figure, in the top right here is a closer look. We have more B cells in the dark zone here and B cells in the light zone as well. So let's take a closer look at this figure. So these are the key events of the germinal center response. Essentially, B cells travel back and forth between the dark and the light zones by these chemoattractant gradients CXCL12 and CXCL13. So these B cells follow CXCL12 to the dark zone and then 13 to the light zone. Starting in the dark zone, we have B cells that undergo proliferation by clonal expansion, somatic hypermutation, and also class switch recombination. After that, the B cells then follow the CXCL13 chemoattractant gradient to the light zone, which is on the right here, and then they undergo positive selection. So the B cells first capture antigen, which are presented on these follicular dendritic cells, or FDCs, and then they are filtered by T follicular helper cells. And so what we're left with is low affinity variants, which are eliminated by apoptosis, and high affinity variants travel back to the dark zone along the CXCL12 gradient to repeat the process. And then after many rounds of this process, with B cells traveling back and forth between the dark and the light zones, we are left with affinity matured B cells that then exit the germinal center to differentiate into either plasma cells or memory B cells. So essentially, our simulation here today models B cells' activity during germinal center reactions, and how and also how environmental chemicals may disrupt this response. And the end goal here is to help us better understand and predict the health risks on GCRs. So the gene network in the germinal center response involves a lot of different pathways, as seen in the top left here. We decided to simplify the network into the one seen on the right, with CX04 and CX05 controlling the migration of B cells in response to the chemotracking gradients CX1212 and CXL13, which is what I just went over. And then we have MYC and AP4, which control B cell growth and proliferation. We also have the MHC2 signaling between the B cells and the T cells, FOXL1 signaling between B cells and FDC, and finally NF-kappa B signaling between the B and T cells. So then this simplified intracellular network was then modeled using tellurium, which I'll get into more detail later. So moving on to the code now, there are currently 4 key aspects in the simulation. The first is modeling the major participating cell types. And here we have 4 types. The first obviously is B cells. Then we have follicular dendritic cells in the light zone, and also T helper cells in the light zone. And then in the dark zone, we have CXCL12 expressing reticular cells, or CRCs. And here in this code snippet here is initializing the cell types in the XML file. The second is B cell chemotaxis, which is dictated by the chemotracking gradients CXL12 and 13 and express receptors. And then the third key aspect is light zone B cell interactions and outcomes, which include B cells capturing antigen presented by FDCs, as well as positive selection by T cells. And then finally, in the dark zone, we have several B cell outcomes, which include B cell proliferation and also somatic hypermutation. So before I talk more about the code, I'm first going to play this video of the simulation, sort of the end result, so that everyone, so that it's easier to understand. So on the left here, we have the CRCs seated in the dark zone. And then on the right, we have the FDCs, which are the larger cells, and the T cells, which are the smaller ones in the light zone on the right. And these cells moving back and forth between the dark zones and the light zone are the B cells. So what these different colors represent is the cell's affinity scores, or basically their ability to neutralize antigens. And the warmer colors in red indicate a better ability to neutralize antigens, while the cooler colors indicate a worse ability. And as they move back and forth, you can see them mutating, so changing colors, and also proliferating in the dark zone. And gradually, their scores start to increase. So we are starting to see some red cells, which indicate the highest possible affinity score, or the best ability to neutralize antigens. If I skip towards the end of the video, it can be seen that there are, one, there are a lot more B cells, and two, nearly all their colors are red, which indicates a super high affinity. Yeah, and here are just some snapshots of the simulation at different time steps. So as time goes on, we have more B cells and also higher affinity B cells. So 5 main files were used in the simulation program. The first is just the main Python script, which was used for running the simulation. The next was the XML script for initializing the simulation parameters, for example, different cell types. The next is the Python separables file for the code itself. The fourth was a PIF file. And finally, there was a separate Python script outside of Computational 3D for exporting and processing data. And we'll be doing some exercises using that code later. So in the XML file, we set up a 100 by 100 2D plane as the boundaries for the germinal center. So we have x equals 100, y equals 100, and z equals 1 because it's a 2D simulation. So when we ran the simulation, we encountered a problem where the B cells would get stuck to the edges of the plane, so near the walls. So we decided to implement our own surrounding wall to help prevent the stickiness. And so here is the PIF file. We have the medium, obviously, and then the walls here are the next 4 lines. And by increasing the B cell wall contact energy values, this eventually helped us prevent the stickiness. So next, moving on to seeding cells. This was done in the separables Python file. Cells were seeded in the start function of the main sepable. And this code shows CRCs. So CRCs were randomly generated in the dark zone by picking random values, and they were restricted within an ellipse centered in the dark zone. So here in this for loop, you can see that 18 CRCs were generated, and then based on a random X value that was chosen, we determined the Y boundaries so that they would conform within an ellipse. And then we would choose a random Y value between the upper and lower bounds, and we were left with an XY coordinate where we could seed the CRC. And essentially, FDC and T cells were randomly generated in the light zone, but using the same method. So all of the FDC cells were assigned a predetermined target sequence because they are the antigen presenting cells. So here in this code, you can see that each FDC was assigned the antigen sequence as a dictionary value. In this case, it was named target. So now onto establishing B cell clones. To make each B cell unique in their ability to neutralize antigens, we had to randomly initialize their antibody sequences. So basically, cells with identical sequences as the target that was just given to FDCs would be the most effective at neutralizing the antigen presented. While on the other hand, sequences with no matching letters would be the worst at neutralizing the antigen. So going back, suppose the target sequence was AGTCT. If the antibody sequence assigned to the B cell matched exactly and was also AGTCT, then it would have the highest possible affinity score and would also be the most effective at neutralizing the antigen. So to do this, we used nested for loops. So this first for loop indicates that we randomly generated 5 sequences, and then for each of the 5 letters, we chose a random letter and appended it so that we would be left with 5 random sequences that were of length 5. So now the B cells could be randomly generated. These B cells were randomly assigned the dictionary value that contained one of the sequences that were previously generated. And then after they were assigned the sequence will be removed from the list so that it couldn't be assigned again. And then after the list was empty, then B cells would stop being generated. So now with all of the major cell types in the simulation, here's what it looks like so far. We have these B cells, which are generated throughout the plane, moving back and forth, as you saw in the simulation video. And we have CRCs seated in the dark zone to the left, and then T cells and FDCs were seated in the light zone to the right. And again, the T cells are the smaller cells, while the FDCs are the larger ones. So to finish initializing the B cells, they were each assigned the same set of parameters. And this included a target volume of 16 and a lambda volume of 4. Chemotaxis was also initialized, and the lambda values for CXCL12 was 1000 and CXCL13 was 0. And so this way, when the simulation starts, the B cells would be traveling towards the dark zone. And also, of course, a tellurium gene network model was added to each B cell. In this case, it was just called Bcell network. So in order to simulate chemotaxis, the chemotaxis plugin had to first be initialized in the XML file, and that's seen in the left right here. And in the right, and we can see that CXCL13 was secreted by FDCs in the light zone, and CXCL12 was secreted by CRCs in the dark zone. And so here's just a look at the chemo tracking gradient fields. The CXCL12 field is here on the top, and the field on the bottom is the CXCL13 field secreted by FDCs. And you can see that these fields correspond to the locations of the cells on the left here. So the CRCs are clustered around the center of the dark zone, and that's reflected in the field on top here. And the FDCs are scattered around the light zone, which is also reflected in the field at the bottom here. So tellurium was used to simulate the intracellular molecular network, and this drove the migration and mitosis behavior of B cells in the model. This screenshot only has the reactions and not the parameters or the initial conditions, but it corresponds with the simplified gene network that I showed previously on the left. So just as like a quick reminder, we have CXCR4 and 5 controlling migration of B cells, FOXO1 signaling between B cells and FDC, NF-κB signaling between the B and the T cells, and finally MYC and AP4, which control the B cell growth and proliferation. So using the neighbor tracker plugin, we determined whether or not the B cell was in contact with the FDC or a T cell. And this made it possible for checking for antibody affinity and making decisions on apoptosis or survival, proliferation, and etc. So using this neighbor tracker plugin, this was first initialized in the XML file. And then down here in the Python code, first we found a list of all the B cells neighbors, and we retrieved that list. And then using the cell type IDs that were previously defined in the XML file, we can determine whether or not a B cell is touching a certain cell type. So here in this example, FDCs have a cell type of 2. So cell type ID of 2. And so we just check if 2 is in the neighbor list. And if it is, then the B cell is touching the FDC. So next, I'll go over the specific cell-to-cell interactions, starting with FDCs here. So the antigen from the FDC is expressed along with the MHC2 antigen on the surface of the B cell by setting the dictionary value to 1. And then we wanted to determine the affinity score. So the affinity score was calculated by comparing the B cell's antibody sequence to the antigen sequence retrieved from the FDC. So there are 2 steps here. The first is obtaining the antigen sequence from the FDC. And as you can see here, the process is pretty similar to the process used for checking for neighbors, except in this case, we retrieve the neighbor cell's target antigen sequence. And then the second step is assigning the new affinity score. And to do this, we start with a new score of 0, and then we compare each letter of the B cell antibody sequence and the antigen sequence retrieved from the FTC. And each time there's a match, 1 is added to the score. So if all 5 letters match, then the score will be 5. And if no letters match, then the score will just be 0. And finally, down here, the affinity score determines the BCR signaling strength by downregulating voxel1. That's this last line right here. Next, moving on to B cell interaction with T cells. Here, we first want to check if the B cell contacted an FTC first, and that's done by using the MHC2 antigen value. So if the B cell touched an FTC, if it came into contact with a T cell before it came into contact with the FTC, which is indicated by the MHC2 antigen value equaling 0, then nothing happens. But if the B cell is touching a T cell after contact with an FTC, which is indicated by this value equaling 1, then all this code here is run. And what this does is essentially positive selection based on the affinity score. So here the percentage chance of survival is determined by the affinity score divided by the max score, which is 5. So a cell with a affinity score of 5 would have a 100% chance of survival, but with a lower score of 2, for example, it would only have a 40% chance of survival. And yeah, so this percentage chance is compared to a random value generated between 0 and 1. And if the B cell does survive, then we turn off the death timer and prevent it from undergoing apoptosis. And the B cell has to travel back to the dark zone. So in order to change the chemotaxis direction, NF-kappa B and CXCR4 are turned on by setting their production rates to 10 and 1 respectively. That's seen here. So the B cell death timer follows a stochastic first order process, and it is initiated by the drop of the NF-kappa B level to 50 or less. And that's this if statement right here. And then once the timer reaches 0, then the cell is deleted, simulates apoptosis. First, the cell is deleted from the gene network, and then the cell is deleted from the simulation. So B cell growth is initiated by AP4 when it is greater than 10. So AP4 is activated by MYC, and MYC is activated by NF-kappa B. And after a B cell comes into contact with a CRC in the dark zone, if the AP4 level is still greater than 10, B cell growth is initiated by increasing the cell volume following this exponential function right here. Otherwise, if AP4 is not greater than 10, then the cell just returns to the light zone by turning off NF-kappa B and CXCR4. So moving on to cell division, if the B cells grow large enough and reach a specific volume of 32 or higher, so recall that the B cells were initialized with a target volume of 16, and so 32 would be double that. The cells undergo mitosis and divide in this mitosis separable class. And then the parent attributes are duplicated to the 2 daughter cells, which essentially clones the original parent cell. So after B cell proliferation and mitosis, they also undergo somatic hypermutation in the dark zone. So if the B cell, if its affinity score is less than the maximum possible score, which means that the B cell's antibody sequence still isn't a perfect match yet, the cell undergoes mutation. So in order to do this, there's 2 steps to the process. First, a random letter of the sequence is selected. That's this line right here. We choose a random index from 0 to 4 since the length of the sequence is 5. Second, we randomly change the letter to a different letter. So we have a list of 4 options here to mutate to. We want to remove the current letter so that it doesn't mutate to itself. And we're left with 3 other options, which we then replace in the sequence that is stored in the cell's dictionary value. So now I'm going to play the same video of the simulation running as before. Hopefully everything makes more sense now. The B cells are going to spawn in the center and they move back and forth between the dark and the light zones. In the dark zone, we have B cells proliferating and undergoing somatic hypermutation. And then in the light zone, the B cells either survive or they undergo apoptosis based on their affinity score. And recall that the color of the B cell indicates the affinity score of their antibody sequence. So a higher affinity is indicated by the warmer colors. And since the simulation just began, most of the B cells have low affinity, but as time goes on here, we have more B cells with higher affinity. So skipping forward, we have some more B cells. We have B cells with higher affinity and also more B cells. And in the left, we're left with many B cells that all have a very high affinity score. So in order to output the data in the simulation for further analysis, we used output text files. And these files were named as, they all had the same naming convention. So it was A_B_C.txt, where A was the generation of the cell, B was the mother ID, and C is the cell ID. And this was very important because new cells were created every time after mitosis. And so we needed a way to link the cell lineages back together. And to do this, we can compare, for example, the mother ID and the cell ID. And if they are equal, then that we know that the 2 files come from the same lineage. And so the data was stored every 15 MCS, which was this save frequency here. And this way, the Computational 3D simulation would run faster, but also our analysis code for plotting would also be faster. Yeah, so here is a visualization of the cell lineages I was talking about before. The first row here are the initial cells, and there were 5 of them. So focusing on one starting cell, 62 here, we can see that every time the cell divided, 2 new cells were created. So after one generation, after one round of division, we have 2 new cells, 74 and 75. And then this continued for a maximum of 10 generations down here, which was basically when the simulation terminated. You can see that there are some other leaf nodes here, and these might be because the cells underwent apoptosis and did not undergo another round of division. So in order to parse and plot all the data from the simulation, we used a separate Python program outside of Computational 3D. And essentially, there were 3 main steps. The first was taking the files from Computational 3D and importing them into Python. The second was finding all of the lineages. So for example, 62, 74, 84, 92 down to 310, that's one lineage branch. We want to find all of those lineage branches. And then the third step would be to select one of those lineage branches to analyze and then make figures. So starting with the text files, here is basically a look at all the text files that were exported from Computational 3D. So A, again, A is a cell generation. So this file right here was for a generation 1 cell. B is the mother ID. So the mother ID of this cell was 59, and the cell's own ID is the third value, which in this case was 64. And in total, there were 267 text files for all the cells. Inside each file, there is a header. So this basically lists all of the variables that were saved for output. So for example, the step, cell ID, in this case, it was all 59 because it's one cell, lambda 12, lambda 13, and it's cut off here, but that says affinity score. A new line was made every 15 MCS based on the save frequency. And the way you read this file is basically going column by column. So column 1 is MCS, which we see is 0, 15, 30, etc. And then column 2 would be the cell ID, and then etc. So next, we imported all of these text files to Python. And these files were just opened using the Python readlines function. So using this this function I made called read files, you can open a file and it will return all of the lines in a list. Next, we used a recursive function to find all of the cell lineages. So each time this retrieve lineage branches function was called, it would check for the children. It would find the two daughter cells of the cell, and then recursively it would run the function on each daughter cell all the way until a leaf node was found. And so if the cell has no more children, then we know that it's a leaf node, and we can append the whole lineage branch to a list. And so here the list to store all of the lineage branches was initialized, and we would run the recursive function on each initial B cell, basically just using a for loop. So the function generated around 130 lists of lineages. And so the output up here are just some of the examples. So you can kind of make out what's going on here. So we know that 59 is a cell that was initialized since the cell ID and mother ID are the same. And then the cell 59 had two daughter cells, 65 and 64. And then the cell 65 had two more daughter cells, 70 and 71. And then 70 had two daughter cells, which were 80 and 81. So yeah, it's very useful having both the mother ID and the cell ID in the name, and the generation as well, obviously. So yeah, so here the first four lineages all went up to the third generation. The next lineage went up to the fourth, and then this one is cut off here, but it went up all the way to the sixth generation. So going back inside the Python code, we select a single lineage branch from the output to perform analysis, and we just copy and paste it here. So to combine all of the cells in our selected lineage branch, we just use a for loop. So here is an empty list to store all of our branch data, and then for each file in our chosen branch, we run the read file function, and we add it to the list of data. So now that we're left with one large list of all the data from one lineage branch, we want the user to choose the desired variables. And so to do this, first we print out a header so that the user knows which variables to choose from. So it's basically just the first line of each file, MCS, cell ID, and everything else. And then the user selects 3 variables to plot. So for example, they can plot the X variable as X center of mass, Y as Y center of mass, and Z as the MCS. And then the user's input would be stored in Python. So now that we know, now that we have all the data for one lineage branch and we know which variables that the user wants, we extract the data. So this is just using list comprehension, but it's essentially just a for loop. So we're going through each row in the branch data, and we're taking the nth index of each row. So let's say, for example, the user wanted MCS for their X variable, then we would choose the 0th index of each row for every single row in branch data. And here we're just checking for whether or not the number is a float, because some lines are headers and we don't want those in our data. Here, Python returns the list as a bunch of strings, so we want to convert them into floats for plotting. And to do this, we just use NumPy, and we just use np.array. And finally, we have all our clean data, and we can plot them using matplotlib, which is a Python library. And here I just have 2 examples of one is a 2D plot and the other is a 3D plot. For the 2D plot, it's just a simple XY scatter plot, and then the Z value will be the color bar or the color map. And then for 3D, it's just the 3D plot. So taking a look at some of our figures that were generated from the simulation. Figure A here on the left is we have the trajectory of a single B cell lineage. So you can read this as if it was a Cartesian like XY plane. And the change in color here indicates the change in time, and that's visually displayed using this color bar. So darker values were indicate that the cell is at the start of the simulation, and lighter values indicate that the cell is nearing the end of the simulation. So we can see that the cell started around here near the edge of the dark zone, of the light zone, and then traveled back and forth until the simulation terminated, and it ended up at, inside the dark zone. Figure B is similar, but instead it depicts a change in the CXCR4 level as time goes on. So we have the X still as X position, but in this case, the Y is the time instead of the Y position. And so here it can be seen that as the B cell heads towards the dark zone on the left, the CXCR4 level increases, which makes sense because the B cell is supposed to follow the chemoattractant gradient CXCL12. And then when it returns to the light zone, the CXCR4 is turned off, and it repeats as the simulation goes on. So here are 3 more figures. Figures A and B on this slide are of sort of the same format as the figure B from last slide. So we have X as the X position and Y as the time. And the Z's at this time are MYC and FOXO1. And it's pretty clear here that FOXO1 inhibits MYC because as FOXO1 increases, MYC decreases. And obviously this corresponds to our gene network. Figure C here depicts the cell generation, and the change in colors here are all in the dark zone because that's where the B cells undergo mitosis, and this makes sense. So there's one division that occurred here, one here, another one here, and another one here. And in total, this was the cell started at generation 0 and ended up with generation 7. Finally, this slide depicts the overall change in antibody affinity. So starting with this this line plot here, we see that at the start of the simulation, there were mostly low affinity cells at around 1, and then near the end of the simulation, nearly all the cells had a affinity score of 5. And that's also shown in these histograms here. So at MCS of 0, we have majority of cells being low affinity, and then at the end of the simulation, nearly all of the cells had a perfect affinity score. And again, this corresponds to our snapshots here. We have relatively low affinity score cells, and then at the end, we have these high affinity score cells. The number of cells is shown here in the top right. The simulation ended up with more than 1,000 cells, and the increase in the number is, again, also seen in the snapshots here. So a low number of cells and a high number of cells. So studies show that the GCR is suppressed by dioxin, which is observed in mice. And so here, dioxin inhibits B cell proliferation. And taking a look at our figures generated from the simulation, we see that the overall ending antibody affinity, they aren't as high. So we still have some low affinity cells. And also the total B cell numbers were reduced to less than 400. So that's nearly one-third, that's nearly a one-third reduction. So in summary, this preliminary CC3D model qualitatively sums up the key events of GCR, and also future iteration is needed to improve this model so we can better understand and predict the adverse outcomes of immune suppressing chemicals, such as the one shown in the previous slide. That's it for the presentation. A big thank you to my mentor, Dr. Zhang, and I do have some slides for exercises after this, but I think now would be a good time for any questions. So.
Any questions for Derek? Feel free to put them in the chat, or if you'd like to unmute and ask, please raise your hand. We can unmute you. We've got a question in the chat from Shalai. How is the affinity score calculated?
Yeah. So if you recall the affinity score, so we have the FTC with a target sequence, which is essentially the antigen sequence, and it's just a string of 5 letters. So for example, A, T, G, C, T. And then each B cell is assigned a antibody sequence, which is also 5 letters, and it can be anything in this case. It can be, for example, TGCAT. And in order to calculate the affinity score, we go through letter by letter and compare the 2 sequences. And if they match, then we add 1 to the score. So if the sequences are identical, then all 5 letters match, and that would be a score of 5. If none of the 5 letters match, then we have an affinity score of 0. So hopefully that makes more sense.
Go ahead. You should be able to unmute now.
Hi. So I think this was an astounding work and a very well-rehearsed presentation. And as you might know, there's a hackathon project after this workshop, which is this weekend. And it is my exact same project topic. So if you're willing to stick around after the session, I'd be grateful for some additional questions.
Sure, yeah, of course. That's amazing.
Okay, thank you.
Yeah, we can be sure to put you in touch too if you don't have a chance to connect today. But yeah, we'll follow up on that. Any other questions for Derek before we move on to the exercises in using this plotting tool? All right, so feel free if you do have a question to just go ahead and leave it in the chat, whenever you get a chance. But I think, Derek, if you want to go ahead and move on to exercises, if we need to, we can stop for a minute and I can explain to people how to get to whatever version of Python is going to work for them.
Great, sounds good. Yeah, so now I have several plotting exercises, which is, again, it's in Python outside of Computational 3D. Yeah, so for these exercises, I'm going to first just go over them really quickly, and then I'll share my screen and do a walkthrough. So after running the Python script, it will prompt you if whether or not you need the lineages, and here you can type capital Y or N. If you type Y, then it prints out all the lineages, and you can choose one. And you basically just copy and paste it into this code and replace whatever list is here. So next, now that you've chosen your lineage branch, you can select 3 different variables to plot. So you can essentially just copy and paste from the header output here, or just type them in, but just make sure that the capitalization is correct, since it is case sensitive. So in this case, I just chose X center mass, Y center mass, and MCS. Third, you can try changing the output from a 2D plot to a 3D plot. And to do that, you just comment out all of this code and uncomment the code for the 3D plot. And finally, we have changing the color map. So the colors, the colors that I used was this first one right here, but you can try any of the other ones, and there are lots more if you search it up online in the Matplotlib documentation. Yeah, so these were the 4 exercises I had in mind. Now I'll just share my screen and do a walkthrough.
Real quick, let's get people set up with Python so they can follow along with you.
Yeah.
So by show of hands, who has Python installed locally? So everybody should have Python installed locally if you installed Compusel locally, because it comes with the package. We also can do this through a Jupyter notebook. I can walk people through how to set that up in just a minute, but we'll get the local install set up first. So if you do have Python installed locally, I think about what you think, Derek, you think using idle is probably the easiest way to
I think
do this. We'll just use the built-in.
Yeah,
IDE.
honestly,
so, yeah, who's familiar with idle? Idle is the built-in interpreter that you can use with Python on the installation. All right, so for those who are running Python locally and know how to use idle, there's 2 things you do need to know. Compusel does not automatically install Matplotlib or, oh gosh, what was the other package?
I think it was Seaborn, yeah.
Seaborn does not install Seaborn, which are 2 things that you will need. Let me go grab this. So you'll need to put in your command line 2 commands. The first one is this, so we'll use pip to install Matplotlib. And Seaborn have an E on the end, or is it just Seaborn?
I think it's just Seaborn.
Yeah. And then you also want to install Seaborn. Now you'll need to run your command line as administrator to install Seaborn. It does require administrator privileges. So for those who are running Python locally, give that a try. You may already have matplotlib from other work that you've done. I think Seaborn is a little bit less common. So for those running locally, we'll do it that way. And then I'll do a quick walkthrough of how to get this set up on a Jupyter notebook for those who don't want to mess around with that. So Derek, let me grab the screen share real quick.
Sounds good.
This will only take a minute. All right, so for those of you who have used Nanohub, which I think is everybody in here at this point, we'll go to the Jupyter notebook through Nanohub. So I'm logged in here. I'm going to go to my dashboard. We'll click Jupyter Notebook. We did, so something actually worth noting here, if you are using Jupyter Notebooks on Nanohub in the future, we found recently that there's a slight bug. Normally you can click this window to just launch the tool from your quick launch bar. For some reason, if you quick launch this Jupyter Notebook with this icon, it takes you to a version of Jupyter Notebook that is looking at the server side storage. So you can't actually make or upload anything. You can, but it goes away when you terminate the session. So you actually have to click through to the splash page and then launch tool this way to get to your account storage. I'm not sure what's going on there, but that was an issue that we discovered a few days ago. Yes, so Edgar has put in the chat, people who are using Mac, the commands I put in are for Windows. People using Mac, please do help each other in the chat. I'm not as familiar with the Mac side of things. So once we launch the Jupyter Notebook tool here, for those following along, what you'll want to do is make a new folder. We'll just call this, so let's see, it made it here. Zoom in a little bit. Go ahead and rename this to whatever you want. I'm going to do module 9.3.1. You'll click through there again. Let me get you a link for the demo code again. So you'll go download the demo zip file from Google Drive. I'll give people a second to do that so that they can follow along here. And then once you have the demo zip file, you will go to upload. I've got it here. And then don't forget, you have to click upload again. Jupyter Notebook likes to trick you that way. And then once you have that uploaded into your folder, now we need to make an actual notebook to use this. So we'll go to new. So staying in this folder that you just created, you'll go to new Python 3.8. And so we'll have a new Jupyter notebook here. First thing I like to do just to make sure that this started in the right place is to run an ls to list the contents of the folder. There's the demo.zip. So to unzip this, again, if you're working on Mac, you will need to rezip it to upload because we've had issues with Mac unzipping things automatically. Excuse me. Oh no, here's my mute button. And trying to hit the mute in a panic before you cough or sneeze is the hardest thing in the world. But you want to upload this zipped because we need to unzip it using Jupyter, which we do simply by !unzip demo.zip. And you do need this exclamation point to run this properly. This is a batch shell script. So we'll run that. And now just to double check that it worked, we'll do another ls. You'll see now we have our demo folder. And then we will cd demo to change into the demo folder. From here, you can just do %run main.py. And this will actually just go ahead and run the script for you. And you'll have to, every time you run it, you'll have to do the run main.py in a new cell again, or you can just refresh this cell. But that was the very quick overview. Anybody have any questions about the Jupyter version of this? Yes, so once you install Matplotlib, the first time you use it, it will have to do some backend stuff to get set up. It only has to do that once, though. I'm going to repost the demo code here for everybody. So go download demo.zip from this folder. And then Julia, you'll get to your Jupyter notebook here and Nanohub. And then you'll want to make a folder in your main directory. And then upload demo.zip to that folder. You have it uploaded, Julia? Okay, great. Now, Julia, the next thing you do is new Python 3.8 in this same folder here. And then what you'll need to put in is, excuse me, exclamation mark, unzip, and then you'll put demo.zip. In that first cell, hit run. And then cd demo to change into the new demo directory folder. And I usually like to do an ls just to make sure I'm in the right place. And then I'll do % run main.py. And you should end up with an output like this, Julia, and anybody else who's following along. Great. All right, sounds good. So I will go ahead and turn things back over to you, Derek. I think that most people have gotten caught up. If you're still struggling with Python, just kind of follow along for now. We can always catch you back up again later. And again, this video is being recorded, so you can always look back later and try this on your own.
All right, thank you, Hayden. That was very helpful.
Yep.
So really quickly, just running this code. It should print out all of the files and then it asks you whether or not you need lineages. So for demonstration purposes, I'm just going to type Y. And it should start printing out all of the lineages. There are quite a few, so it might take some time. And then now you can choose to copy a lineage into the code, into the chosen branch. So whatever lineage you want. I'm just going to stick with the one I have right here. And then I'm going to select some variables to plot. So to start, I'm going to have the X center of mass, Y center of mass, and the MCS. And here's our plot. We have the same plot that I showed in the results section of the presentation. So the cell starts in the edge of the light zone and then travels back and forth. And now I can either rerun the program and choose a different lineage, or I can stick with the same lineage and change which variables I want to plot. So I'm going to run it one more time. This time I'm going to type no because I don't want the lineages to be printed again. And I can just choose 3 variables. I'll choose the X position, the MCS, and then the generation. And it's working pretty much as intended. So switching over to a 3D plot now. I'm just going to uncomment this code. And then comment out the 2D plot code. And then I can just save it and run it again. Choose the same variables, XCOM, YCOM, and then MCS. And now we have a 3D plot. So pretty cool. And you can drag it around and see different angles. Yeah. Now I'm going to try changing the color map. So I'm going to try changing it to plasma. And all I need to do is replace this right here. And then save and rerun. And now it's a different color. And there are more colors can be found with a quick Google search online, or just looking through the matplotlib documentation. Now I'm going to change it back to a 2D plot and try a different lineage. So I need to run it again to find the lineages. So I'm going to choose a shorter one. Maybe the cell underwent apoptosis early. So all I need to do is copy the line here. and replace this list. Now I can save and rerun. This time I don't need the lineages, and I can plot. And it's slightly different. So I guess the cell started around the same location and then stayed in the dark zone for the majority of the simulation. Yeah, that's pretty much all of the demos I have. Feel free to try it out yourself. If Python isn't working right now, you can try it on your own later, and feel free to reach out if you have any questions. So.
Yeah, so this is a great, so the code is available to you in the demo folder that we shared. This is a great template for people to use for visualizing some of their own data. Obviously, it will require some modification to fit your own purposes. But please do take a look at the code at some point if you're interested in these kinds of script-based plotting tools, because we think that this could be transformed with a little bit of effort into something useful for a number of different types of projects. It's also a good way to get some experience with Python. Modifying a template to fit a new purpose is always a good way to practice.